So we've gone through the process of transcription, right? We started with DNA, we used RNA polymerase, made our mRNA strand. But if you're a eukaryotic cell, something else has to happen before you want that mRNA to just leave your nucleus. We go through a post-transcriptional processing process, right? Basically, we're finishing up, making it look kind of nice and pretty and make sense. We're gonna do some mRNA splicing, we call it, where we edit out introns, and then we're gonna make our mature mRNA transcript by adding a poly A and a GTP cap to our spliced mRNA molecule, or our mature mRNA molecule. First, I want to talk about actually our poly A tail and our GTP cap, because they're a little bit simpler, okay? Basically what happens, this mRNA is gonna move through the cell, right? It means it's gonna move through the cytoplasm, and there are enzymes that can come and kind of attack it and break it down, and we don't want that to happen. So we added GTP cap and a poly A tail to the ends of it just to kind of protect it and keep it whole. The poly A tail is anywhere from 100 to 200 adenine nucleotides at the end. It increases stability and it helps with exporting from the nucleus. The GTP cap happens at the 5' end and it's modified guanine nucleotides. It's going to protect the transcript and it's actually going to help the ribosome attach to the mRNA when it gets to that point. So I like to think of them kind of just as protection, helping them get where they need to get. So they don't get destroyed basically along the way. Another thing we need to do is we're going to remove some non-coding portions of mRNA. And you're saying, what? I just coded for a gene. Doesn't the gene carry all the important information? Don't I need all of that? Well, actually what we found are there are these parts called introns and exons. Exons are the part we need. Exons, I say, exit the cell. Get it? So like they're the parts you want you to leave. Introns, however, we actually kind of splice out. We used to call them junk DNA, but we've kind of found out that they're not really junk. They're important, even if they're not really like used in a specific mature mRNA molecule. So what we say though is introns are mRNA sequences that do not code for amino acids. So basically, we cut them out and they're not included in our mature mRNA. However, exons are the sequences that code for amino acids. So they're retained during RNA processing, this finishing up part, if you will, and they're going to go ahead and leave as mature mRNA. We call this splicing when we cut them out. And this picture is kind of nice, right? Here's our big strand. We have our poly A tail, our GTP cap. We're going to cut out our introns through splicing, and we get our mature mRNA. Looks kind of like this. It's shorter than our original, if you will. How do we do this? Well, actually we do it with these things called SNRPs or spliceosomes. They're small nuclear RNA plus some proteins. They kind of identify the spot. And then we have a ribozyme that's actually going to be responsible for cutting out that chunk. Then what we can do is actually pretty neat, something called alternative splicing, right? So we can splice at different points in a gene, meaning an intron isn't always an intron, right? Even if it's spliced out in one instance, it might actually be used in another time. So different segments can be treated as exons under different conditions. Different mRNA transcripts can be produced from one primary transcript, which is cool. We get more diversity, right? Diversity is good. So here's an example. Let's say here's my gene. I have exon 1, 2, 3, and 4. Okay, I make my mRNA sequence, or my pre-mRNA, sometimes we call it, looks complete. Looks just like my gene from my DNA, right? Well, through splicing, I can either make mature mRNA that has like 1, 2, and 3, or it could make mature mRNA that has 1, 2, and 4, right? So they're differences. They're going to go on and make probably different polypeptide chains, which will make different proteins, which will probably have different functions, which is cool that we can get all of that out of the same transcript. So that's alternative splicing. We get alternatives based on how we splice. Just in summary, some things to think about. Transcription in prokaryotes versus eukaryotes, right? Prokaryotes, DNA is in the cytoplasm. It's circular chromosome. We say it's naked DNA. It's not enclosed in any sort of nucleus, and there's no introns, no processing afterwards. Eukaryotes, right? DNA is in the nucleus, it's in linear chromosomes, and it's really tightly around these proteins called histones, and it has these introns and exons, so we have to go through this processing even after we've gone through transcription. I hope that was helpful. I hope you can walk yourself through the process of transcription, and now in our next video, we're going to start to see where does that mRNA go to be translated.